Today we are talking about a subject that's quite dear to my heart. This is the inner ear and central auditory pathways. I guess everyone's got to have their favourites. I spent years studying cochlear physiology, and so this is my chance to tell you all about the amazing things that the cochlea does. The title says the inner ear, but to be fair, I'm not going to talk very much about the vestibular system. So the peripheral vestibular sensors are also part of the inner ear. They're the posterior part. We're going to be focusing on the anterior part, which is the cochlea, which does the job of detecting sounds that are in the environment, converting them into neural signals, electrical signals that can travel up the auditory nerve. And then we'll spend a little bit of time about talking about where it goes from there. What happens to those signals that go along the auditory nerve? How do they get processed by the brain? And that is part of the central auditory pathways portion of the lecture. Are there any questions before we get started? No? You're all good? Excellent. Let's go. Well, jumped ahead a bit. There we go. All right, so the inner ear consists of the cochlea, which is the organ of hearing, and the vestibular organs, which are the organs of balance. So just very, very briefly, the vestibular part consists of these three semicircular canals. Their job is to detect rotational acceleration in all three planes. So if you move your head left and right, up, down, it's those receptors which are doing that main sort of detection of, it's very important to know where your head is in space, interestingly. Partly it's to do with balance. But also, very importantly, it's to do with your eye movements. It's sort of like a built-in Steadicam for your eyes, which is amazing. So if you move your head up, down, left, right, just focus on a point on the board, move your head up, down, left, right, your eyes are compensating exactly for that movement so that the image stays fixed on your retina and doesn't blur. So if you get a camera and just go like this, you're going to get an awful result. So this is it's basically a survival reflex to help you focus on things while you're running through the savannah being chased by something. All right, so the inner ear is embedded in the temporal bone that you can see here. This is one of the major bones of the skull. This little hole here is actually your ear canal that we mentioned. All right, so this is what my inner ear looks like. This is a pair of 3T MRI. T is Tesla, which is the strength of the magnetic coil. How did I get these? I wasn't ill I wasn't needing an MRI or anything, but you can see here it says CMRF Open Day. So the Canterbury Medical Research Foundation had just bought a brand new MRI machine, and they said, we need someone to lie in the machine for a couple of hours while people stroll through and go, wow, look at that MRI machine. And I said, yeah, I'll do it, but you have to get me good scans of my inner ear. And so they did. So all it cost me was just lying on a table for an hour or two, which is great. So they didn't do a very good job of cleaning it up. There's little bits of floaty stuff. But you can clearly see 1, 2, 3 semicircular canals. And this part over here is the cochlea. And that's the bit that we're going to be focusing on today. All right, so what does the cochlea do? Essentially, it transforms the sound-induced vibrations of those middle ear bones. We've spoken about that whole process of how it gets to there. So down the ear canal, moving the tympanic membrane from the malleus to the incus to the stapes. The stapes footplate fits into the oval window of the cochlea. So the cochlea has two windows. It's like play school. The oval window and the round window. And then behind that is the cochlea, which is full of fluid. So we take the vibration of sound, which is in air, and we've got to somehow get it into the fluid of the inner ear, and we do that through that impedance matching process that the middle ear does. And within the inner ear, that's where the transduction of that sound into electrical signals takes place. I'm going to use this word transduction. I might have mentioned it before, but a transducer is something which converts something from one modality to another. So an example is the microphone that you can see here. So this is a transducer. It is converting the air pressure signals to an electrical signal in the wire. Does that make sense? Does everyone get that? So that's a transducer. What's the opposite? What takes electrical signals and turns them into sound pressure? Speakers. Absolutely. So those are both examples of transducers. So it comes from the Latin word trans, meaning to cross, and ducare is like to lead. So it's like if you lead someone from one side of the river to another, that's transducare, and this is a transducer. So the inner ear is essentially a transducer. All right, how well does it do it? Amazingly. So to do this job, it meets specifications not yet achieved by any human-designed system. So it's sensitive to sub-nanometer displacements. That's a billionth of a meter. It's ridiculous. It has a response time in microseconds. It operates over a million-fold range of intensities. So from the threshold of hearing to the threshold of pain is a ratio which is the same as 1 mouse to 5 elephants. And we're able to cope with that massive, massive range of sound pressures. It has a frequency range of 20 hertz to 20 kilohertz. That's 10 octaves. So if we think about a normal piano, there it is, 88 keys. The human ear extends it way down here to 20 hertz and way up there to 20,000 hertz. So that's how long your human ear piano would be. I could probably not hear these end ones here because I'm older than you. It's just a fact of life. It can detect intensity changes of 1 decibel. And it detects frequency changes of 0.1%. So just by comparison, adjacent keys on the piano are 6% apart. And that means between every key, you can fit another 60 keys. Like, you imagine the size of that piano, particularly with all those extra octaves. So it's a ridiculously sensitive device. So, and all of that contributes to our ability to extract signals in noisy environments. And I'll talk about that in a bit more detail later on. It's particularly the outer hair cells which help with that job. And the brain, of course. So not surprisingly, there are lots of things that can go wrong, and at least 85% of hearing impaired people can trace their problems to the cochlea. So here are some rows of hair cells. We're going to talk a lot about hair cells today. These are the inner hair cells, and there's one row of those. They have fairly straight stereocilia, and here's 3 rows, 1, 2, 3 rows of outer hair cells, and they have sort of a V or W-shaped stereocilia. We're looking down on the top surface of the organ of Corti, which is the name, it's named after a dude, Alphonse Corti. And that is sitting on top of the basilar membrane. So the basilar membrane is a long membrane which stretches from one end of the cochlea all the way to the other. It's like a trampoline, very springy, and it's a tuned device. So it's got a higher mass and stiffness at one end than the other, and that means that it vibrates preferentially to some sounds at one place and different frequencies at another place. So we have a high frequency end of the cochlea, which is near the base where the stapes is, and then extends all the way to the low frequency end, which is the apex of the cochlea. The far end of the cochlea is where the low frequencies are processed. So here's what it should look like. Lovely, straight, aligned rows, but loud noise, ototoxic drugs, old age, all of these things can contribute to the death of hair cells. And once they die, unfortunately in humans and other mammals, they don't get replaced. So we've been working on it, I say we, they, have been working on replacing hair cells for 30, 40 years, and we're getting closer, we're putting together all the pieces of the puzzle. Animals like chickens, avian hair cells and bullfrogs and all of these other creatures can just, need some more hair cells? Here you go, have some more hair cells. They'll just regenerate them, but unfortunately ours don't. So they just get replaced by supporting cells. So it looks a bit like a desert in some places. And what that means is that, particularly with the inner hair cells here, if there are no hair cells in this region, you can't hear that sound with those hair cells. But if the vibration gets big enough, neighboring hair cells can pick it up, but then it will sound like 2 sounds that are not quite the sound. And they interact in a really horrible way, and it sounds scratchy and distorted. Okay. So the inner ear was named the labyrinth by the Greek anatomist Galen of Pergamon in the 2nd century AD. You can imagine getting lost in those, do I turn left, do I turn right? And it consists of the cochlea that you can see here and the vestibular part, and it's a bony shell, which we call the osseous or the bony labyrinth, and contained within that are a set of membranes. We call this the membranous labyrinth. The point of the membranes is to separate 2 special types of fluid. One of them is very similar to physiological saline. So what's the main salt in physiological saline? Sodium chloride? Yeah, excellent. So that's just basically table salt. So it's a variation of that. But the other fluid, which is within these ducts, so within them, so you've got the bone, then you've got the sodium chloride solution, and then within the tubes is a special one, which is potassium chloride. Quite different, and that behaves totally differently, and that powers this whole process. All right, so let's focus on the cochlear part. If we zoom in and chip away that bony shell, we get a view of what's going on inside. This is not a human cochlea. As I'll show you in a moment, our cochlea is a bit squashed and flat. But in some other mammals, it's quite elongated. So guinea pigs and that sort of thing have quite a taller looking cochlea. But here you can see, here's the oval window where the stapes footplate pushes in and out. Every time the stapes footplate pushes in on the oval window, the round window here comes out a bit. So it's like push, pull, push, pull. Can you imagine that? Push, pull, push, pull. And what it's doing is sending vibrations into these the fluid. So what we have are, as we go around in this spiral, it's like a spiral staircase going around and around and around. And in fact, we actually call them scalae, and that's the Latin and Italian and I think Spanish words for a stair, is a scala. So this one here is called scala tympani, and it goes around like this. This one here is scala vestibuli, which goes around, and you can't see it here because it's been damaged by the freezing process, but there's a membrane that crosses over here, and this little chamber in the middle is called scala media. Media in this case meaning in the middle of those other two, so scala media. And that's the one that contains the potassium concentration in there. And it goes all the way around to the very top, and then there's a hole at the very top which is called the helicotrema. All right. So here it is. Here's another view. This is the bony shell has been completely removed, and we just get this nice view of the basilar membrane spiraling around. You can see the distribution of frequencies here. High frequencies down in the base at 20K, and then we move around, the frequency that these places best respond to changes till we get down to very low frequencies at the apex. We can take the cochlea and stretch it out, straighten it out, and that helps us visualize what's going on. And you can see here's the stapes, this is the base of the cochlea, this is the high frequency end. Can you see how narrow the basilar membrane is? And when we get to the low frequency end, it's a lot floppier and a lot longer. Just like on a piano, you've got the short strings and then the very long strings and so on. Does that make sense? Cool. All right, so when we put in sounds of a high frequency, again, this is grossly exaggerated, this image. It's really like an atomic shimmer. So it's like atomic dimensions, but we magnify it tremendously so you can actually see what's going on. When we put in a high frequency sound, it vibrates the basal end. That's the adjective related to base. The base, basal. And this is the apex. What's the adjective for that? Apical. So we have the apical end, which is the low frequency end. And so you can see low frequencies preferentially vibrate that bit. And can you see how the wave is going like this? It's a traveling wave, like a... the reason it's doing that is because the phase at every single point on this basilar membrane is slightly different to the phase of the region next to it because it's got a different mass and stiffness. Different mass, different stiffness. The phase changes gradually along, so the whole membrane doesn't go up and down like this all at once. It goes in a traveling wave. And it was von Becquerel, the Hungarian physiologist, who discovered that. He won the Nobel Prize in 1960 for his imaging of this traveling wave on the basilar membrane. All right, here is a rather beautiful image. Again, grossly exaggerated, but showing the idea of this traveling wave moving from the base around to the apex. So it sort of does that kind of motion. So actually, the neurons that are, we'll talk about the nerves in a moment, the neurons, they are attached at all points along this basilar membrane, but the ones at the high frequency end actually get stimulated a little bit before the low frequency end. So if you imagine all your nerves firing, let's say we put in a clap sound which stimulates the whole basilar membrane, it contains all frequencies. The nerves don't go like that, they fire in a kind of way, which is funny. Because when it gets back up to the brain, that process is undone. And so we don't hear, we hear like that. So the delay at this end is undone at the other end. It's remarkable. Okay. Here is a three-dimensional reconstruction of a human cochlea. So this is based on CT scans, and you can fill in a computer afterwards, fill these different compartments to get a visualization of what it looks like. And you can see it's a lot more squat than the elongated example that I showed you earlier. And you can read through this at your leisure, but the different compartments are color-coded according to what they are that you can see there. All right, so let's take this cochlea and slice it down the middle. This is kind of like what we would see. So there's bone, obviously. Within the bony labyrinth, the bony shell, is this membranous labyrinth that is divided into 3 chambers, 3 scalae. And they are wrapping around in a, like going up, you know, when you drive into the car park at Litchfield Street or whatever, you go around and around and around. It's that same idea. They're going around and around this spiral. So we've got these 2 scalae here, scala tympani and scala vestibulae. They're on the outside, and they are full of perilymph. So the name of the fluid is perilymph, and that's the sodium chloride solution. It's kind of similar to sodium to physiological saline. So why perilymph? Peri means on the periphery, on the outside. So perilymph is on the outside. What's the opposite of the outside? What's the word for inside something? Like if you want to look inside someone, you use an endoscope. Endo, yeah. So this is perilymph, and in the middle is endolymph. And that's the one that's the high potassium. That's the special juice. So this is sodium chloride. This is physiological saline. It's not super different from the other fluids that are around your body, but this one is almost unique. And it's generated by a very special tissue, which is located on this lateral wall here that you can see there. And it's highly vascular, this part here. You can't see it in this image, but it's basically red. It's this red stripe that goes all the way around because it's got the highest, like a super high blood supply, and the highest metabolic rate of any tissue in the entire body. It's doing a remarkable job of creating this high potassium solution and pumping it against its will into this chamber here. So it uses a lot of energy to do that. And because it's red, here's a shell. Its Cochlear comes from the word, similar root to cockle shell. It means it's a shell shape. Anatomists, again, with their imagination. Imagine you can actually see this red stripe from the outside, and it's got the name Stria vascularis, which just means bloody stripe or vascular stripe as it goes around the top. Okay, here's another look at a cross section. Let's zoom in on one bit and focus on the membranes of interest. We have to separate these different juices from each other by means of a membrane that things can't cross easily. So the first one here is Reisner's membrane, and that separates scala vestibuli from scala media, and it's pretty boring. So we used to think it was just like glad wrap, but we now know more. It does do some stuff. There's very little in nature which doesn't do stuff. Or doesn't multitask in some way. So it does do stuff, but for our purposes today, let's just assume it's pretty inert and just sits there separating those 2 different fluids. The complete opposite of boring is the basilar membrane. That's this one here. This is absolutely astounding. So as I said, there's a gradation of mass and stiffness from one end to the other, so that it's basically doing like a Fourier transform mechanically from one end to the other. Different parts of it are vibrating as you go. Have you ever yelled into a piano? No, there's actually a tiny room next door with a piano. So maybe if we can get in there, we can go and yell at the piano. But what you do is you open the lid, and if you go, and stop, you hear the piano going back to you. Try that, because what you've done is you've set it resonating, and that resonance continues. After you stop, it continues for a bit. That's essentially what the basilar membrane is doing. You put in energy to get it moving, and it keeps moving a bit, but it doesn't ring out forever because it's in fluid. So this whole environment is in fluid. What's it like running through the water? Is it as easy as running on land? No, because you've got to push through all the fluid. And it's the same with this whole environment here is in fluid. So there's a lot of friction, a lot of damping. We need to overcome that in order to hear properly. And the way we overcome that is via these hair cells on the outside here. These are called the outer hair cells. That's their job, is to overcome the friction and make it not like we're in water, but like we're running along the beach. Now we can go as fast as we want. And that does amazing things to our hearing that we'll talk about in a moment. All right, let's zoom in again. What we can see here are 3 rows of outer hair cells. How many rows of outer hair cells are there? 3. Excellent. And here's 1 row of inner hair cells. How many rows of inner hair cells do we have? 1. Excellent. They are actually slightly different shapes. So the inner hair cells are kind of fat-bottomed. They're like a flask. We call them flask-shaped. So can you see here? Flask-shaped. And the outer hair cells are kind of cylindrical, like a test tube. So they have different shapes. The other thing that's quite different about them is the nerves that connect to them. So this part here is called the spiral ganglion. Do you know what a ganglion is? It's basically where, so when you've got nerves going from somewhere to somewhere else, they've got two main projections. Have you done any neuroscience yet? Yeah? So the axon and the dendrites, yeah, so they often point in different directions, right? And the cell body, the soma of the neuron, is in the middle. In a lot of structures, all the cell bodies hang out in one place, and we call that a ganglion. And this here is a ganglion, but because the structure goes around in a spiral, so does the ganglion. So it's a spiral ganglion all the way around, and every single hair cell on this basilar membrane, so look, let's count. 1, 2, 3, 4, 5, 6. So someone did, and we know exactly how many hair cells there are. There's like 12,000. Every single one of these outer hair cells is innervated by a whole bunch of neurons, like 30 neurons. And they have different jobs. They're not all like 30 copies of the same type of neuron. They've got some ones that are really, really sensitive and will fire at the smallest provocation, and others that wait until, I'm not going to fire until it's a big signal. And so they all split the job between them about encoding quiet sounds versus medium sounds versus loud sounds. It's really amazing. But 95% of these neurons go to the inner hair cells. Look at all of this innervation of these hair cells. 95% of them go there. A measly 5% cross this area here called the tunnel of Corti and make contact with the outer hair cells. Only 5%. So already that tells you that the main job of the outer hair cells isn't to send neural signals to the brain. If that was important, they would have a lot more neural innervation. So their job is something else, and we'll talk about that in a moment. So what's the job of the inner hair cells? What are they doing? Sending signals to the brain. And that's obvious just by looking at how many neurons are connected to them. All right, let's look again at these cochlear fluids. Exactly where are they? So we said the one on the outside, the perimeter, the outside is the perilymph, and I'm showing that here in yellow. And the one on the inside is endolymph. And that's the one that's high in potassium, that is produced by this special tissue here called the Stria vascularis. This is it here. In Latin, the bloody stripe. It's a 3-cell layer pumping epithelium. It just means it's like sheets of cells, and they have this amazing job of, we extract some potassium here and we pump it. We like pushing it uphill, pushing it uphill, pushing it uphill, get it in there, and the scala media saying, no, we're already full of potassium. It's like, never mind, have some more, and it just shoves it in there. And because, what's the charge on a potassium? What's the valence of potassium? Is it negative ion or +1? It's a positive ion. It's already quite positive in here, and we're shoving more potassium in there, so it gets even more positive, and that creates a battery. So an AA battery, what's the voltage on an AA battery? You guys not read? Do you not read the labels on your batteries? It's 1.5 volts, right? 1.5 volts. What about the ones that are square with the two, when you stick your tongue on them, they zing? Yeah, that's a 9-volt battery. If you've not licked a 9-volt battery, go home and do it tonight. So 1.5 volts. This here is 95 millivolts. So that's like, an AA battery is 1,500 millivolts, and this is like, almost a tenth of that. So this is a pretty big voltage for a physiological, biological tissue to produce. So this is high in potassium, and it's got a really, really ridiculously large positive voltage. Okay, we've got 3,500 of these inner hair cells. As I said, their job is to convert the vibration signals into electrical impulses that go along the auditory nerve to the brain. Okay, are you ready? I'm about to show you the outer hair cells. Here they are, 3 to 5 rows of outer hair cells, normally 3. There's about 12,000 of them in each of your ears, and their job is to amplify the vibration of the basilar membrane by up to 1,000-fold. So imagine the sound coming in. We're surrounded by, we're on this trampoline, but we're surrounded by water, and so it's really hard to move. But the outer hair cells come along, and they actually vibrate in time with the signal and make it 1,000 times bigger. You've all been on a trampoline, presumably. Anyone here not? Just blink rapidly. No? Have you ever been on there with someone else? Yeah, all the trampolines these days say, be safe, only one at a time, but it's actually no one ever does that. So if you're on there with someone else and you time your bounce just right, you can fire them up into the air. I'm sure you've all done that, right? The double bounce. That's literally what these guys are doing. Their vibration is timed so that they are making that vibration a lot bigger. Again, this is ridiculously exaggerated, but look at the way that that is increasing the vibration of that basilar membrane. The cell is actively changing its length every single cycle. Did you know cells can get longer and smaller actively? So they're actually just going like this. And that makes the vibration 1,000 times bigger. This is, as I said, exaggerated. Look at the hair bundle at the top going. In reality, this movement is ridiculously small. So take just this blue bit, these stereocilia, and scale them up to the size of the Sky Tower in Auckland. At the threshold of hearing, imagine the very, very, very tippy tip of the Sky Tower moving by 3.5 centimetres. That's all it has to move for us to be able to hear that hearing. That's pretty cool. All right. Can you see that these stereocilia are kind of embedded in the green thing that's on top of them, and that's helping push them left and right and left and right. They're kind of getting shoved like this. So here's the stereocilia, here's the reticular lamina, which is this red line. As I move up, watch what happens to the pen. Can you see how it's bent over that way? And now what happens when I move it in the opposite direction? It's pointing at me. So I'm the center core of the cochlea. It's going away from me, towards me, away from me, towards me. So an up-down movement of the basilar membrane turns into a lateral medial movement of the stereocilia of the hair cell. And that does some really cool things. We'll talk about it in a moment. So this green thing, its only job is to sit on top of the hair cells and provide something to push against that. So it's like a veranda made of jelly. It's an acellular matrix. It doesn't have any cells within it, doesn't have a blood supply, it just sits there touching the tops of these hair bundles, causing them to move backwards and forwards as the basilar membrane goes up and down. All right. And then the signals that are generated by the inner hair cells, they release neurotransmitter. They are basically modified nerve cells. They are releasing neurotransmitter onto the primary afferent neurons. What is an afferent neuron? What does afferent mean? Goes to the brain. And what's the opposite of afferent? Efferent, and they come from the brain. Does the cochlea have any efferent innervation? Is there any reason the brain would need to send signals to the cochlea? In fact, it does. It has a pair of volume controls, which is pretty amazing. So we've got two types of efferents that go down there. There's the medial efferents there. They come from the medial superior olive. They come down here and they say to the outer hair cells, whoa, 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 turn it down a bit. And then you've got the lateral olive, which sends the lateral efferents. They come down and attach to the nerves just here, and they just turn down the volume a bit. So it takes you further away from threshold. So if you ever had that ringing in your ears where, so not tinnitus that many of us have all the time, but just sometimes you'll be sitting there and suddenly, and it'll be there for 60 seconds, you'll have a feeling of fullness, and it will just die away over a minute. You've all experienced that? Many of you have. That dying away is your brain saying, hang on, what's this? What's it? Let's turn that down. That shouldn't be doing that. And it does that via efferent control. It's pretty remarkable. Okay, so these primary afferent neurons, they're the 95% of them go to the inner hair cells. They send signals up to the brain, and that becomes part of the cochlear portion, or the vestibular cochlear nerve, which is the 8th cranial nerve. We'll talk about that a bit more later. All right, so to summarize, outer hair cells embiggen the vibration by a factor of 1,000. That makes it much easier for the inner hair cells to detect that sound. What is 1,000 in decibels? We're going back a couple of lectures here. Count the zeros. Count the zeros times it by 3. No, 3 zeros times it by 20. So 3 zeros times 20, 60 decibels. So that's improved our hearing by 60 decibels. So right now I'm talking at about 60 decibels. So if you didn't have those, my voice would be like the quietest sound you can hear. So it makes it much easier for the inner hair cells to detect it. They detect it, release neurotransmitter onto the nerves, which sends the signal up the eighth nerve to the brain. All right, there to there to there. Okay, so those hair cells come in all different shapes and sizes. This is a range of different hair cell sizes of different mammals, going from 10 micrometers down the bottom there, which has yep, right down there. That has a frequency that it is best able to detect. This is in kilohertz of 160,000 hertz. What's the highest frequency humans can hear? 20,000. That can hear 8 times as high as we can hear. So what animal would need that? What animal can use ultra-high, ultrasonic sound? Bats. Yeah, guinea pigs do hear very high, 40K, but bats use it for echolocation. That's how they can fly around in a dark cave and not smack into walls. They can actually beam out ultrasonic signals. They get a reflection off a moth in the air and they go, aha, there it is, and they can swoop in and grab it, all in pitch dark through this echolocation. All right, humans are these guys here. So we range from this size, which can detect 20,000 hertz, to the very long ones, which get down to around 20 hertz. We have some other animals. This is a bat, as I said, and the mole rat, little guys that just crawl in the dark through tunnels. They can hear very, very low frequencies, and they have these very long hair cells that you see there. So they carry out what we call mechanoelectrical transduction. Remember I said a microphone is a transducer, speakers are transducers. Mechanoelectrical transduction is converting a mechanical signal into an electrical signal. So in human, as I said, there are about 15,000 hair cells in each ear. How do we know? Because we counted them. All right. So those hair cells, the hair bundles at the top, are the things that give the hair cells their name. Another word for them, they kind of look like cilia that you get on other, in the gut, for example. They look like cilia, but they're not cilia. So we don't call them cilia. We call them, they're kind of like cilia. We call them stereocilia. That's what it means. So stereocilia. And if we look closely, we can see 1, 2, 3 rows. We've got a short row, a middle row, and a tall row, and each of these rows is connected to the next tallest row by a tiny, tiny little string. Can you see this here? See that little tiny thing there? That is called a tip link. Tip link, and here's an electron micrograph view. You can see it spiraling around like that. It is minute. When the hair bundles move backwards and forwards, when they're in the direction of the shortest stereocilia, can you see how this all gets slack? Can you see it's all slack and bendy? And then when we move it in the opposite direction towards when we bend the hair bundle towards the tallest stereocilia, it pulls tight on that elastic and it gets nice and taut. So it's just basically like this guy here. So short, and then when we deflect in that direction, can you see how that's stretching? That's putting pressure on this end and this end. And at the ends, that pressure is used to open a little door. So imagine like our door over there, we can open it up. Imagine I've got elastic tied to that, and if I pull on that, that door opens, and when I loosen it, the door shuts. Does everyone get that idea? That's an ion channel. They are real things. They open, they let a particular shaped object through, or a particular charged object. You can be very selective about what it lets through. In this case, these channels say, if you're negative, you're not coming through. That should be the motto of this class. Yeah, no, you're only positive. A positive ion, what's the name of a positive ion? A cation, excellent. I always like to think of cations as being positive, because people like cats. And anions are negative because onions make you cry. That's a terrible metaphor. So these are cation sensitive channels. You pull open the door, only positive ions can come through. The two positive ions that are there and able to flow in, there's calcium. Calcium isn't the big guy in this story, it's potassium. That potassium that we worked so hard to pump into the scala media above us, that is the thing which floods into the hair cells, comes through these little ion channels that are located in the stereocilia, and it flows down the stereocilia into the body of the cell. All right, how small are these stereocilia? Look, here is a single red blood cell, and you can see the size of it. I mean, this assumes that you know how big a blood cell is. Anyway, the message is they're very, very tiny. So here's these tiny stereocilia. All right, so look at this. At rest, when the hair bundle is leaning this way, the door is shut because there's no tension on the spring. But when we pull in the other way, look at this, it yanks open this ion channel and inflows our positive ions, calcium and potassium. And it's potassium which does all the work in this job. Can everyone see that there? Anyone got any questions? All right. Okay, so imagine, you can see how small these are. These are the very, very delicate parts that can just bust like that. So ototoxic drugs, loud noises, old age can just break these tip links, and you don't replace them. Okay, so how and why, when we open the door to potassium, what is it that makes potassium want to go through it? It doesn't want, obviously, but what is it that forces potassium to go through that door? There's a few things. So this is, I did this as part of my PhD. So these are all of the channels that are contained within the outer hair cells. And part of my PhD was creating a mathematical model of how they all interact. So each one of these channels had its own equation. You could calculate the net voltages and then calculate the movements that would ensue and how it can regulate itself to keep our hearing pristine all the time. Like when you bend over to tie up your shoelaces, you don't go deaf, which is handy. So this is the top of the hair cell, and you can see there are these stretch-sensitive mechanoelectrical transduction channels. And at the bottom, there's a whole other bunch of potassium channels. So the potassium goes in through the top and out through the bottom. So we've got the displacement-sensitive potassium channels at the top, and then this bunch of channels down the bottom here. All right, so here it is. Where did the potassium come from? What's the tissue that's pumping that potassium? The bloody stripe, the Stria vascularis, that's pumping the potassium into there. So now we've got a heap of potassium and a positive voltage. We call that voltage the EP, the endocochlear potential, plus 95 millivolts. The inside of the cells is actually quite negative. So the outer hair cells are minus 70 millivolts, and the inner hair cells are about minus 40 millivolts, slightly different voltages. So if they are, let's have a closer look. We've got the outer hair cells here, inner hair cells here. Here's the reticular lamina. We look across there. So we're going to look in this view like that. This is what we would see. There's the reticular lamina. Here's our outer hair cell. Here's the stereocilia. There's the jelly awning veranda on the top. When I move the hair bundle up like that, can you see how that has pulled on the spring and it's opened the channel? Yeah, so closed, open, closed, open. So here it is closed, there it is open. So just like, imagine you're filling the bath and you go shh, 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 shh with your hand on the tap. So the water is flowing, then it's not flowing. Then it's flowing, then it's not flowing. All right, so the potassium here is hanging around. Is potassium a positive ion? Yep. So do positive things like to hang around with positive things, or do they try and move towards negative things? They are drawn electrostatically towards the negative things. It's just like magnets, the north and south ends. This is super positive up here, and that's super negative down there. So if I give them the chance and open the door, where are they going to go? They're going to go down their electrical gradient. So they go in through the top and into this hair cell. So potassium flows into the hair cell because it's a positive ion, and it's super negative down there and super positive up here, so it flows down into it. That's cool. What happens to the voltage inside here? If this is super negative, and suddenly I dump a whole bunch of positive ions in there, is it going to make it more negative, or is it going to undo that a bit? It's going to make it a bit more positive. Yeah, so if a cell is very negative compared to its environment, we say it's polarized. It's polarized. And if I make it less polarized, I call that depolarizing. So I've just depolarized this a bit by adding a bit more positive ions through there. The potassium flows out the outside, and instead of going down an electrical gradient, because it's actually still very negative here, and it's 0 millivolts out here, so it'd be going uphill electrically, there's so much more potassium in here than out here that it's flowing down its concentration gradient. So that's the other thing about ions, is they move from high concentration to low concentration. And there's like an exchange rate of what's more important, the concentration gradient or the electrical gradient, and there's equations to figure that out. And in this case, it's in favor of potassium moving out like this. So it flows down its concentration gradient into that, into the outside world down there. Not the outside world, the Corti lymph, which is a variety of perilymph. So to summarize, here we go, is everyone listening? We move the hair bundle back and forth. That opens and closes the channels at the top, like putting your hand on the bath and going shh, shh, shh. Hair cells are negatively charged. They're polarized. And if we dump some potassium in there, potassium is positive and it makes the hair cell less negative. We call that depolarizing. And when we move the hair bundles in the other direction and close the door, less potassium is flowing in. So what happens to the voltage inside the cell? It goes a bit more negative again, because we've got less potassium coming in. So this is amazing, with every single cycle of the sound, condensation, rarefaction, condensation, rarefaction, the eardrum is moving forwards, backwards, forwards, backwards. The malleus, incus, and stapes are moving forward, backward, pushing in and out on the oval window of the cochlea. That's making the fluid in the cochlea have more pressure, less pressure, more pressure, less pressure. That makes the basilar membrane go, what happens first? It goes, if you pull back, it goes up. If you push forward, it goes down. So it's going up, down, up, down, up, down. Every time it goes up, the hair bundles move in the direction that opens the channels. Every time it goes down, it moves in the direction that closes the channels. So if I'm listening to a 1000 hertz sound, the hair bundles are moving backwards and forwards 1,000 times a second, and the voltage is going up and down 1,000 times a second. And if we do that at 20 kilohertz, it's happening at 20,000 times a second. The voltage is going up and down, up and down, up and down. So that's a signal that the hair cell can use. Yeah?
Is that the reason why, or is it connected to the reason why when people get hearing aids, they get so tired from wearing them? Is it a physical tiredness
No.
because they're hearing new sounds, or just a like...
Yeah, it's a fatigue because so when something's fantastic and autopilot, you're not thinking about it. Have you ever driven home and you get home and you go, oh, I'm home. Like, I forgot that I've actually driven all of this way and was paying attention the whole time, but it was really your brainstem driving you home. You've forgotten all about it. You're not using all that effort. But let's say you're in a different car or you're driving in the US and you're on the wrong side of the road. You are like paying so much attention to not make any mistake, right? So that is using all of your mental effort because you're dealing with something that is different. So it's the difference between what's automatic processing, what's requiring conscious effort. And so we've only got a limited amount of cognitive resource at any one time. And that's why, let's say if you've got chronic pain or if you've got chronic dizziness or anything like that, it messes with your ability to think about other things. So it all impacts. And so if you're struggling getting used to a new hearing aid, you're actually applying a lot more cognitive power towards that processing. And that's why you feel exhausted at the end of the day. It's just like you guys. You're learning new stuff right now. You're going to walk out of this lecture going, like a real physical tiredness. Okay, so this whole process is mechanoelectrical transduction. We're turning a mechanical movement to an electrical signal. That's pretty cool. Hey, all right. So I'm not going to talk about this slide. You can read it, but this talks in detail about exactly all of the steps I just mentioned in a little bit more detail. And basically that's what we just did. So the voltage inside the cell, when we're not doing anything, it's a particular voltage. I said it's minus 70 for the outer hair cells, minus 40 for the inner hair cells. We call that the resting potential. That's just what it does in silence. But when we wiggle the hair cell backwards and forwards, it goes from being a DC resting potential to a wobbly potential, an AC potential. AC means it's alternating. It goes up, down, up, down, up, down, up, down, up, down, around that DC level. And we call that the receptor potential. Potential just means a voltage. Receptor potential means the voltage in there because it's acting like a receptor. So each of these hair cells, the outer hair cells and the inner hair cells, when the stereocilia move back and forward, they each have a cyclical voltage signal. Like it's like sinusoidal, but it's distorted. Gets a bit flat on the tops and bottoms. But each of them, the voltage inside the cell is going up, down, up, down, up, down. And that says, hey, there's sound present. Does that make sense? So when there's no sound, the voltage is like this, but in an area that is got sound in it, let's say I play a 1 kilohertz tone, your 2 kilohertz hair cells are just sitting there. But your 1 kilohertz hair cells are going like this 1,000 times a second. So what do the inner hair cells and the inner hair cells do with the voltage that they have each got? Have we established that they have different jobs? They look different, they are different shapes, they have different degrees of neural innervation, they have different machinery. What do they do with those voltage signals? Do they do anything different with them? The answer is yes. Here we go. Inner hair cells and outer hair cells both have stereocilia. Potassium enters. It makes the voltage go up and down in exactly the same way, but they do something different with the voltage signal. Inner hair cells use that AC receptor potential, the wobbly voltage, to control neural firing. So every time they depolarize, what happens to a nerve when it depolarizes? Let's say a nerve terminal. Depolarize a nerve terminal, it releases neurotransmitter. You're looking quite blank. I've got some slides on that later on that will help remind you. When the voltage gets a bit more positive, it releases neurotransmitter to stimulate the next nerve along, right? So inner hair cells, the voltage goes positive, negative, positive, negative. The inner hair cells go, they only fire on the positive going voltages. They send neurotransmitter out when the voltage gets positive, and they don't when it gets more negative. So they're going, That's true along most of the cochlea, the low frequency end in particular. They have cycle by cycle firing. In the high, don't tell anyone, but in the high frequency end, when there's a sound, they just go. That's important, but beyond the scope of today's lecture. The outer hair cells, the voltage goes up, down, up, down, up, down. When the voltage goes up, the hair cell goes, and when the voltage goes negative, it goes like this. So they are literally changing their shape cycle by cycle 1,000 times a second. How on earth do they do that? How do they convert a cyclical voltage change to a cyclical movement? I've taken a voltage and turned it into a movement. Do you remember we called that process before? This is mechanoelectrical transduction. I take a mechanical signal, turn it into a voltage. The outer hair cells are doing that, but they're also doing the opposite. They're taking that electrical signal and turning it back into a mechanical signal. They are jumping up and down on the trampoline. We call that electromechanical transduction. So we've got MET, MET, and this is EMT, which is the reverse of that. That's the outer hair cells. They're the only cells that can do this, the outer hair cells. How do they do it? There's a protein called prestin that is embedded in the lateral wall of these cells. So here's the cell wall. It's got a matrix to give it structure. And embedded in there is a whole heap of this particular protein, which is a transmembrane protein. In a former life, it used to be a chloride transporter, but it's mutated and can now change its shape. How did they discover this? They looked at all of the genes expressed in outer hair cells and subtracted away all the genes that are also expressed in inner hair cells. And they had a candidate, like that whole bunch that were left over. It was like, well, this is something that outer hair cells do that inner hair cells don't do. And they, one by one, put those proteins into like kidney cells. And then they put this one in, and the kidney cells began to move with voltage. And they said, hey, presto, that's it. And that's why they called it prestin. But do you want to see the hair cell move with a voltage signal? So this square wave here is a voltage signal. And this pipette is a tiny glass micropipette which is stabbed into the cell and can change the voltage in the cell, right? It's a way of controlling the voltage in the cell. Look at what the hair cell is doing. It is wiggling backwards and forwards. Every time it goes positive, the hair cell contracts. Every time it goes negative, it expands. Cycle by cycle, it is contracting and expanding. And imagine that sitting on the basilar membrane. It's jumping up and down. It's got three brothers right next to it. Well, two brothers. There's three of them. I don't know why I've assigned them a gender. Never mind. And there's lots of them all side by side. They are all doing that in unison. Can you imagine what that does to the vibration in that region? That's pretty incredible. Jonathan Ashmore, lovely guy, he's at UCL in London, and he got on the news with this video because he took that voltage signal and said, let's make it a bit more interesting. And he played music to the hair cell. And this got around the world, and now everyone's got this mental image of my hair cells are dancing along with music. But here it is here. Might be a bit loud. Anyway, there you go. So that is pretty remarkable. What effect does outer hair cell, we call that the active process. We're using an electrical signal. It takes energy to do that, right? So cut off all the energy, the cell's just going to sit there. But it uses energy, puts energy back into that vibration. We call that the active process. It's from the outer hair cells. We call that the outer hair cell active process. What does that do to our hearing? It does a number of things. Should we take a 5-minute break and come back to this? Yeah, excellent. So you know where the toilets are? Excellent. We'll see you back here shortly. Okay, so we've established that outer hair cells are miraculous and amazing and fantastic. because they can twitch in time with the thing and they make the vibration 1,000 times bigger. Is that all they do? What's the point of making the vibration bigger? Well, obviously it helps us hear what that vibration is. So what effect does that have on our hearing? Is it just like turning up the volume knob? The answer is no. It is far more important than that. So I mentioned that moving the stapes backwards and forwards puts sound into the fluids. So we've got sound in the air right now. The stapes going backwards and forwards puts that sound in the fluid. So now we've got sound in the fluid, just like we've got sound in the air. That sound will have a variety of frequencies contained within it. If one of those, I'm going to say this is the 1 kilohertz place on the cochlea. So I'm going to say that's the 1 kilohertz place. If I put in a 1 kilohertz tone, that 1 kilohertz place will be the one that vibrates. Okay, it's not going to vibrate for other frequencies. It's going to vibrate for 1 kilohertz. That's a lie, actually. So any lower frequency will cause that to vibrate. So we call an upward spread of masking, and I spoke about that in the masking lecture. But let's just for today say that that's the one that's going to most vibrate. 1 kilohertz will most vibrate this portion right here. And we get this traveling wave that goes like that. It grows and it peaks at exactly this place, which is the 1 kilohertz place. So we've got this relationship between the place and the frequency. Place, the word for place is, we say topic, and tone is for frequency. So we call this a tonotopic organization. Each frequency has its own place. We have a place frequency map, a relationship between the frequency and the place. So which is the high frequency end? What do high frequencies vibrate? Yep, this base, the basal end up here. And where do the low frequencies vibrate preferentially? The apical end. Excellent. All right. Cool. So let us have a closer look at this region here. For a very quiet sound, we get a small vibration. And for a loud sound, we get a tall vibration, right? But imagine we didn't have outer hair cells. Are we in air running down the beach, or are we running through the water of the ocean without outer hair cells? What are we doing? We're through the water. There's a lot of friction. And when you've got a resonant system and you add friction, it dampens the tuning. So here is my water bottle. It is full of air. Sorry, it's full of water. And if I go like that, it kind of, sound, you can hear it, right? But if I was doing this underwater, it wouldn't make that dinging sound. It would make a bleh sound, right? Because it's not sharply tuned anymore. So without outer hair cells, this is what your tuning would be like. But when we add outer hair cells and they sharpen, they bounce up and down, make the vibration larger, it actually sharpens the tuning. And look how tuned it is now. It's vibrating with this massive, massive peak located over this tiny bunch of hair cells. So imagine, do you think things would sound better if we're stimulating just the right hair cells, the 1 kilohertz hair cells, or is it fine if we stimulate everything from 980 hertz to 1030 hertz, all the same? Is that fine? No, I like to use the analogy, it's like playing the piano with your fingertips, ding, 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 and playing with the flats of your hands or with your fists, right? Which one sounds better? You haven't heard me play. No, it's, the fingertips, right? That is the equivalent of what we are hearing. That's why outer hair cells and protecting your hearing is so important. We aren't just making sound louder. That's why hearing aids don't miraculously restore your 18-year-old hearing, right? When you have a hearing aid, it makes the vibration louder, but it doesn't make your tuning sharper. The only thing that can make your tuning sharper is having outer hair cells that cancel friction, I'm sorry to say. So outer hair cells make this vibration 1,000 times larger, but also look how sharp it is. It's ridiculously sharp. And that helps us tell the difference between sounds that are very close in frequency. Otherwise, they're all mushed together. It's like taking a photo, but it's blurred, right? You've got no detail. But if you have sharpness of tuning, it's like a clear, in-focus photograph. So the outer hair cell active process puts energy back in there. It undoes the effect of being in the water. So now we're like running on air. It cancels friction. It provides negative damping. It cancels the friction. Increases the vibration by 40 to 60 decibels and sharpens that tuning. how does that correspond to the relationship between how much sound we're putting in and how loud it seems, or the sensitivity or vibration of the cochlea? Imagine I put in sound at 50 decibels and I get this minimum level. Let's say this is the minimum level of sound that of vibration that is needed for your hair cells to detect it and fire off signals to the nerve. This blue line is without an active process. As I make it louder, it grows in a linear way. So this is a straight line, decibels and decibels, that's a straight line. We call that the linear growth of vibration. If I make it a bit louder, the vibration gets a bit louder. But if I add outer hair cells, so here's your threshold, the louder sound, the sound has to be 50 dB in order for you just to detect it. But if we add outer hair cells, look what it does. It boosts your sensitivity for low level, for quiet sounds. It makes it easier to hear quiet sounds. So now your threshold is down here at what we've decided to call 0. Can you see how much extra hearing you've got because you've got outer hair cells? Is that like turning up the volume on your stereo? Is it? Is it like, so if I turn up the volume on the stereo, does it make the quiet sounds louder and the loud sounds louder? It does, yeah. This doesn't do that. Your outer hair cells make the quiet sounds louder, but look what it does to the loud sounds, 120 dB. It does nothing to them. So this makes your vibration, the growth of loudness, non-linear. Non-linear. It's not a line anymore, it's a bendy thing. And what that means is quiet sounds get amplified. The outer hair cells do heaps of work and amplify the quiet sounds. But if sounds get beyond a certain level of vibration, they're like, oh, I'm doing all I can, but I'm not going to make this any louder. And so we have this growth of loudness that is normal after that point. Not normal, it's linear. So we've got this non-linear region and linear. So where are the outer hair cells helping? Quiet sounds or loud sounds? Quiet. And they're not amplifying loud sounds. That's super important because our whole brain assumes that this is what's happening. Our whole brain has calibrated itself so that, oh, I'm expecting this red line. So that when sounds get to be a certain loudness, oh, I know how loud that is. Do you know what happens when you remove the red line by damaging your outer hair cells and you've just got the blue line? Your sense of loudness goes like this. You say, my mouse is here. I go, can't hear it, 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 can't. Oh, I can just hear it, just hear it, just hear it. Whoa, too loud. You leap from I can barely hear it to it's too loud in such a short amount of time, level. So we call that difference between the quieter sound you can hear and the louder sound you can tolerate. We call that your dynamic range. So without outer hair cells, you have a 60 decibel dynamic range. That's the range over which you have all of the difference between can just hear it to, wow, that's really loud. It's squished into 60 dB. But when you have outer hair cells, that pushes it out to 120 decibels. That's why our audiogram goes from 0 to 120, right? So that's our dynamic range, 120 decibels, because we've got outer hair cells. Lose the outer hair cells, and you lose that dynamic range, particularly in the high frequencies. The high frequency end needs extra help because that's the bit that is stiffest and needs more boosting. The low frequencies don't tend to need as much help of outer hair cells. So if I wipe out, let's say I wiped out all of your outer hair cells, what would your audiogram do? So just the outer hair cells, not the inner, they are different, right? So I'd wipe out your outer hair cells. Would your whole audiogram go thump? No, it's helping the high frequencies more. So your audiogram would go, from your perspective, it would go, ugh. The high frequency end would fall. But the low frequencies could hang around a bit. They would go down a bit, but not as much. So that's why it's such a common audiogram configuration is that high frequency sloping hearing loss. Because that's where the outer hair cells are doing their job, is at the high frequencies and for quiet sounds. Does everyone get that? Fantastic. All right. So look at this. Here's your beautiful fingertips. This is the tuning curve showing these particular hair cells are tuned to that frequency. If I get rid of your outer hair cells, no longer are we playing the piano with our fingertips, we're playing with our fists. Look how rounded that tuning is. This is with damaged and missing hair cells. And if we lose the inner hair cells as well, we get dead regions, which shift all of our thresholds up. So we go way up. We've got to, across the board, we've got to make everything louder. All right. that can give rise to some weirdo results perceptually, right? So when we take an audiogram, do you know what we're actually doing? When we play you a sound that's the quietest sound you can hear, I'm actually touching the very, very tip of the tuning curve of the hair cells in that region. So this tuning curve here, see this one here, this very tip is your threshold at that frequency. This is a guinea pig, not a human. That's why we're measuring up to past 20 kilohertz. This is the tip of the tuning curve, and our audiogram is just all of those tuning curves upside down. It looks like this. So if I am playing you a 3 kilohertz sound, I'm touching just the 3 kilohertz hair cells. They're the ones that are most tuned to that region, right? But look what happens if we have acoustic trauma and we damage these particular cells. Look at this. This isn't just like a pyramid, or an upside down V. It's got a low frequency tail. So if I make a low frequency sound loud enough, I can stimulate these other frequencies. Low frequencies can stimulate higher ones. So if I damage this 1 kilohertz hair cell and its tuning curve goes down and down and down, this 10 kilohertz one has escaped that trauma, and its low frequency tail is actually now more sensitive than the tip of that 1 kilohertz sound. So if I play you a 1 kilohertz sound and it's very, very quiet and I make it louder and louder and louder, when you first hear it, in this case, this poor person isn't hearing it with their 1 kilohertz neurons. What neurons are hearing it? The 10 kilohertz ones. How does that sound? That must be awful. So that is called diplacusis. It means two places. We're playing you a 1 kilohertz sound, but you're picking it up with your 10 kilohertz neurons. So everything sounds distorted and scratchy and nasty. And that's what can happen if you have particular types of acoustic trauma. All right. Yeah, the 8 kilohertz neurons are responding before the 1 kilohertz, even though we're playing a 1 kilohertz sound. All right, now let's get on to the ascending auditory pathway. The final step in cochlear transduction is the release of neurotransmitter from the inner hair cells to stimulate the neurons below. Inner hair cells are basically like modified neural cells. They have all the machinery to release neurotransmitter, just like a neuron does. And that sends signals up the vestibular cochlear nerve, the 8th cranial nerve. Just to remind you, I know you're all experts on this, but these are some bits of neurons. We have the dendrites. They basically take inputs into the neuron. The cell body is called the soma. Sometimes you have a single long projection on there, which is like the out direction. What's that? The axon. The axon has this fatty covering with little gaps in it. What's the fatty covering called? Myelin sheath. What are the little gaps called? Little nodes of Ranvier. They help speed up that conduction. They make it super fast because the signal jumps from point to point to point to point to point, and it goes super fast. And at the end, we have this stubby bit, the bouton, the axon terminal. That's where the release of neurotransmitter occurs to talk to the next neuron. Tiny gap there called a synapse. Yeah, excellent. Very good. Okay. So here's a beautiful picture from Wikimedia Commons. I really love this. This is great because it reminds you that a nerve cell is a cell. It has all of the things that cells have. It has a nucleus. It has mitochondria. It's got all of those things. It's a cell. But it's got these highly specialized components there. All right. So you're all familiar with the idea of an action potential. Is that right? You've heard that term before? No? Okay. So an action potential is a way of... So what happens is the way the signal propagates down the nerve, it's not like a voltage gets bigger over here and that bigness is just like a global thing that spreads all the way down it. So it's not like I've got a knob that controls the voltage and the whole cell's voltage goes up or it goes down. And I can encode a tiny change or a big change just with the graded voltage. It's not that at all. What it is, it's a little triggered event that when we get past some critical threshold, so the nerve cells are negative, they're polarized just like hair cells are, they're really negative. When we get to a certain threshold, let's say minus 55 millivolts, so they're lower than that, if I do anything to make its voltage go a little bit higher, it'll go back down. And if I go a little bit higher, it'll go back down. But if I hit this magical voltage of minus 55, you know how I said that that's a stretch opening door, that channel? I pull on it and it opens. You've got voltage gated channels where that's got a high charge on it. And if I make an electrical charge here, that's going to attract the door towards me and open it. So if I make the cell more positive, this negatively charged door is going to open in my direction. That's going to open it. So when the inside of the cell gets to minus 55, that magic door opens, lets a bunch of sodium in, and suddenly, bang, the voltage of the cell rockets up to plus 40. So it just shoots up. And then other channels close, other channels open, and it drags us back down again. So when we reach that magical threshold, we get this bang spike in the voltage. And it's that spike which is doing all of the work. It's an all or none response. We can't have a small action potential from a single nerve. When we hit that threshold, we will go to the full size and then back down again. So that's an action potential. Does that gel with everyone's current theories? Yes, excellent. All right, so we have this massive peak. Bang, it's an up-down thing. How do we encode then a tiny signal versus a loud signal? Or, you know, I'm talking in sound terms, obviously, but it's the same as just touching or pushing really hard, right? So we have two things. One is the spike rate code. So the bigger the signal, that firing, like that, it goes from being to and fires super rapidly. So the bigger the signal, the faster that firing rate will go. We call that spike rate code. The rate increases for a bigger stimulus. Because that's how quickly we can overcome this undershoot. For a big stimulus, this undershoot gets smaller and smaller, and those peaks get closer and closer together. The other thing that happens is that a large stimuli, so I said a 1 kilohertz tone stimulates exactly the 1 kilohertz place, but if I make that louder, it actually starts to stimulate the neighbors as well. That spread gets bigger and bigger and bigger, and now I'm stimulating more bunches of neurons from adjacent regions. So I have more neurons actually firing. So that's called the population code. Those two things happen at the same time. Is everyone happy with that? So that's how we can tell the difference between a small signal and a large signal is which neurons are firing, how many neurons are firing, and how fast are they firing? What's the rate of their firing? All right. And then we have synaptic transmission between the hair cells. Sorry, I'm using this diagram. It's got French labels, but it's such a beautiful diagram. So now we have le vésicle synaptique and glutamate, the termination présynaptique. And I apologize for my appalling accent. But anyway, imagine, so this is a neuron, right? But this could just as easily be an inner hair cell. It does exactly the same thing. When the voltage gets positive in here, it releases little packets of neurotransmitter which cross the synapse and stimulate the postsynaptic neuron, which sends the signal up to the brain. So everyone happy with that? Inner hair cells are basically like the presynaptic neuron in a normal synapse. They do the same thing. And they do happen to use glutamate as their neurotransmitter. Okay, does anyone need any more detail on this, or is this all coinciding with your... If anyone needs any more help, I can talk to you after the lecture. All right. So I love this, this drawing here. Here is a single inner hair cell. Look how many nerve fibers are attached to this single hair cell. It's desperate to capture every bit of information it can from that single inner hair cell. There's a bony shelf that runs along here called the osseous spiral lamina. So osseous just means bony, so it's a bony shelf. And look, there are tiny little holes on here that the nerves pass through. That's called the habenula perforata. I love this. So we have the nerves, these dendrites are coming through here, and here's the cell bodies living in the spiral ganglion in Rosenthal's canal. And then the axons head off down the 8th nerve to send the signals to the brain. Everyone see that? Cool. What's another word for the ear canal? The external auditory meatus. All of these nerves head off to the brain through something called the internal auditory meatus. So that's another hole, another little channel which goes off to the brain, but that's where the nerves are heading from the cochlea to. And look, just to give you an idea of the scale of this, look at just the sheer quantity of neural innervation. That's amazing. And then you've got this measly 5% of the fibers actually cross the channel. They take a basalwood turn and head this way, and each one of these little fibers contacts with multiple outer hair cells. And it does that for housekeeping reasons. It sort of says, you know, if we get a really loud, you know, have you ever heard something that's painfully loud and you go, oh, you know, that you get the actual pain sensation? People are hypothesizing now that that's what these neurons are doing. They're the ouchie neurons. Or you could call them type 2 afferents. It's up to you. All right. So all of the nerves from all of the regions of the cochlea, from the high frequency end down here at the base, all the way to the low frequency end, all of those outer hair cells and inner hair cells spiraling around on the basilar membrane. That row of inner hair cells that goes around and around, each one of those has all of those neurons attached to it. And if we start at the top of the spiral, those nerves have got to leave the cochlea somewhere, and they come down the middle of the spiral. So we call that middle portion of the cockle shell, shown here in green, we call that the modiolus. So the modiolus is coming down the middle, and we start at the top. So the low frequency fibers are coming down the modiolus, and then they are joined by the frequencies that are a little bit higher than that. So we have the mid frequencies. They wrap around the outside. And then when we get to the high frequency end, those fibers are wrapping around. So can you see now we've got this fiber bundle leaving the modiolus, but it's actually got an arrangement whereby the low frequencies are in the middle because they started first, then they're wrapped in the mid frequencies, and they're wrapped in the high frequencies. Can you see that? So if I take a cross section of this auditory nerve as it leads, I've got low frequencies in the middle, mid frequencies, outside of that, and then wrapped around the edge of the high frequency fibers. Isn't that cool? One weird side effect of that is there's actually a type of tumor that can affect your auditory nerve. Does anyone know what it's called? It's got two names. It used to be called an acoustic neuroma. So acoustic because it's the acoustic, it's the auditory part of the nerve, and neuroma because it's a tumor on a nerve. But we since now know that it's actually more likely to happen on the vestibular part of the nerve, and it's more to do with the Schwann cells that wrap those nerves. So the proper term is now a vestibular schwannoma. Both are used. Acoustic neuroma, vestibular schwannoma, you will hear both. Those nerves, those tumours grow. So they don't spread through the body and metastasize and give you liver cancer or anything like that. They are a tumour, but they just grow, they take up space. So that's how they cause problems, is just by being there and being big. And they grow very slowly. So a lot of old people will die with them, not of them. They are so slow growing, sometimes you don't have to do anything about them, but sometimes they grow when you're younger and you do need something like a surgery to take them out. But one of the ways you know it's there is because they are taking up space in that tiny auditory canal, the internal auditory meatus. They're on the vestibular nerves, which are right next to the cochlear ones, and they grow, and they're going to start pushing on the cochlear nerve. So when I start growing and pushing on the cochlear nerve, which fibers am I going to compress first, the high frequency ones or the low frequency ones? The high. Why? Because they're on the outside. So this tumour's going to push on the high frequencies fibers first. And so what do you think you might see in an audiogram if someone's got a tumour on that nerve? Would you get a hearing loss? Yeah. Where would it happen? In the high frequencies. Is it going to happen in both ears at the same time? No, it's just one. Most of what happens to your ears happens to both at once. Loud noise, both ears at once. Ototoxic drugs, both ears at once. Old age, both ears at once, obviously. But tumours and things like that will usually affect one ear at a time, unless you've got something like neurofibromatosis. And so if I get a unilateral sensory neural asymmetry, that's what we call a red flag as an audiologist. That means, let's look into this a bit closer and refer you off to an ENT for a proper investigation. Does that make sense? So only a couple of red flags that we have, that's one of them, a vestibular schwannoma. All right. So this is where the signal goes once it leaves the cochlea. So this here is the cochlea. What are these things over here? That's the vestibular parts, the semicircular canals, and here is the internal auditory canal. There's the cochlear nerve coming through here. And it follows this kind of pathway. It crosses to both sides of the brain through multiple brainstem relay centers and then heads out to, what's the outside of the brain called? The cortex, heads out to auditory cortex. Auditory cortex lives around here in the temporal lobes, right? If sound goes in my right ear, which cortex does it stimulate the most? The left. Can you see I've made this line a lot fatter than that line? So it crosses over and we primarily stimulate the contralateral auditory cortex. But on the way, it's passing through all of these brainstem nuclei. Here's another view. So the signal goes, we've got the spiral ganglion here. It goes through the ventral cochlear nucleus, goes through the cochlear nucleus, superior olive, lateral lemniscus, inferior colliculus, medial geniculate body, out to auditory cortex. It goes ding, 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 and at each stage of that pathway, different things are happening. They're doing different jobs and extracting different features. So we send all of the auditory signal in, and that gets to the cochlear nucleus. And from there, right from that lowest part of the auditory brainstem, it's throwing information away and extracting features. And it's sending the features up the line. So we're getting more useful, precise information the higher you go up, to the point where what gets up to the cortex is like the very essence of what you need to know about the sound being present or absent. Does that make sense? Timing, duration, spread of frequencies, all that sort of stuff makes its way to the auditory cortex. But do you know what is remarkable? Absolutely remarkable. Remember I said all of these inner hair cells respond to different frequencies? Everyone got that? We've got low frequency hair cells and high frequency hair cells. They all have neurons attached to them. So we've got the low frequency neurons and the high frequency neurons. At every point in this, we call that a tonotopic organization. At every point in this ascending auditory pathway, that tonotopic representation is preserved. So we have low frequencies and high frequencies within the dorsal cochlear nucleus. In the inferior colliculus, you can scan across low frequencies to high frequencies. Get out to the medial geniculate, low frequency sheets, high frequency sheets. We get out to auditory cortex, low frequency strips, all the way up to high frequency strips. So there's this one-to-one mapping of frequency to frequency, all the way up this auditory pathway. So you've got literally a part of your auditory cortex. This is the bit that responds to 1K, and over here is the bit that responds to 10K. They're adjacent, they're near each other. Isn't that's cool? So this tonotopic organization is, oh, I was just explaining vestibular schwannomas, but here they are. Here they are, so you can get a view of it. Here it is growing on the superior vestibular nerve. It's pushing on the facial nerve here, which also, that's the 7th cranial nerve that travels in that place as well. And here it is here, but here it pushes on the cochlear nerve, and that's where you get that high frequency hearing loss. Okay, so auditory perception, we've got all of these, any sensory system does this, but we're going to talk about the auditory one. It has this idea of feature extraction. So we are pulling out different really interesting parts about the features of the stimulus. So by contrast, we're able to detect edges of sounds. So there is sound here, there's no sound at these frequencies, this is where that sound starts. We have a localization of function. So individual cells respond to certain features or little groups of cells. So for example, you might have some cells that respond not just to 1 kilohertz, but 1 kilohertz between 30 and 40 dB. And then if it's 40 to 50, that's a different cell doing that job. So you've got this really specific localization of function. And the idea that it's a hierarchical processing. So we have the raw input comes into the cochlear nucleus. Each step is not dealing on the raw input anymore. It's doing more with the step that came before it. So it's, oh, you've given me this processed, I'm going to process it a bit more, pass it on to the next guy, they're going to process it a bit more, and so on up the chain till we get up to the auditory cortex. So we can divide this whole system into the brainstem. That's where we get the emergence of parallel pathways. We're crossing to both sides of the auditory brainstem. And binaural processing, we're able to compare the inputs between ears. That's super important for things like directional hearing and timing estimates. In the midbrain, we put it all back together, create an auditory image, and then when we get up to the forebrain, the medial geniculate and the auditory cortex, that's where we have the integration with other modalities like vision. That's super important. So if I clap my hands and you heard a sound, you imply that like your brain realizes, oh, it was the clapping of the hands that created the sound. And worse than that, not worse than that, it's very, very cool, but I'll show you a video of something called the McGurk effect. Have you all heard of that? Yeah, so that's where what you see fully determines what you hear. So your visual input modifies your auditory input, and that determines whether you think someone's saying pa or ga or all those things. I'll send you a video because it's very cool. All right. As I mentioned, tonotopic organization on the basilar membrane, low to high or high to low, is preserved at every subsequent layer all the way up. We have all these different regions of the cerebral cortex. You are familiar with all of those. Our dude is the temporal lobe over here, in particular auditory cortex. But the signals from there also then go to the language processing areas, which you guys are going to learn a tremendous amount as well, such as Broca's area, Wernicke's area, all of those different gyri and cortices. All right. But if you've got a stroke in a particular area, that will obviously affect your ability to turn the sounds that you hear into a language percept and be able to understand, extract information about the language that's coming to you. I want to talk very briefly about cortical plasticity. Brain plasticity these days is taking on a different term because of microplastics in the environment. We actually have a tremendous amount of plastic in our brains, like actual plastic. It's frightening. Look it up. We're not talking about that. Plasticity in this case means the ability to change inputs over time in response to different stimuli. So if you take, do you remember I said tonotopic organization? The low frequency fibers in the cochlea go to the low frequency parts of the auditory cortex and high to high and mid to mid. Before, let's say there's nothing wrong with the cochlea, this is the spread that we get, high to high, mid to medium, low to low. If I damage the high frequency part of your cochlea, the low frequencies are unaffected, the medium frequencies are unaffected, but the high frequency part of the cortex now has no input coming to it. Can you see how that happened? Yeah, I've destroyed that part down in the cochlea, so nothing's coming to it. So what happens? It's really a case of use it or lose it. Like if the high frequency, real estate is expensive. So if the high frequencies aren't being used, the medium one is going to spread into there. And so we end up with the medium frequencies are now taking up even more of that real estate and are getting processed by this area that was just sitting dormant, right? What do you think happens if I didn't destroy your high frequency end, but you had a hearing loss and I gave you back a hearing aid to give you back those high frequencies? What do you think would happen? Well, in the first step, so this is before I give you a hearing aid, this area is being processing the medium signals. Then I give you back the high frequencies, and things sound absolutely awful. Oh, scratchy, can't tolerate it because the area that's supposed to be processing them is busy processing those medium frequencies. So there's this 6-month period where a hearing aid sounds atrocious and awful and people hate it, but you have to persist. And that's the thing we always tell our clients is, it's going to sound scratchy and tinny, but I need you to keep wearing it as much as you can. If you wear it all day, over time your brain adjusts. And pretty soon, the high frequencies will be stimulating the high frequency area again, and things will sound a lot better. I am now wearing glasses. If I show you videos from previous years, I'm not wearing glasses all the time, but now I'm wearing glasses, and they're progressive lenses. So they're the ones that have a different focal length down here for close things, and up here is different. When I first put them on, it was the weirdest sensation because everything was magnified down here, and my whole perception was warped. And now, everything is fine. I know exactly where everything is and everything's in focus and my brain is coping. But when I take them off, that's now reversed. Now I'm feeling like things are wrong. And that's because my brain has had those plastic changes to adapt its visual cortex to the new inputs that are coming in. All right, in the last half an hour, we're going to talk about how our auditory nervous system helps us to localize sound. And have I spoken about this a little bit already? I have. We'll go into a bit more detail. So we are humans and we can tell where sounds are coming from in 360 degrees all around us. It's useful for us, but it's particularly important for animals like barn owls who have just like the most amazing skill at this. So they'll sit in a tree and there's a snowy field in front of them and a little field mouse will just like make a little tiny rustle under the snow just over there and they'll go, right, and dive down and just pluck it out of the ground. They know exactly where it is because they have the most remarkable directional abilities. Absolutely amazing. So we can do the same thing. And the way we talk about, so how do you give a location on a sphere around you, any of these directions? Firstly, you can say, what is the azimuth? So this is, let's say that's 0. And then I can go around 90 degrees. So that's 90 degrees azimuth. 180 is behind me, 270 back to 0. Does everyone get that idea? So that's this direction, the horizontal plane. For each of these horizontal planes, I can go up, horizontal directions, I can go up and down. And we call that the elevation. So I can go up 90 degrees or down 90 degrees. Does everyone get that? And every combination of azimuth and elevation gives me a point on that sphere that I can localize a sound to. All right, so we've got a number of different clues for helping us do that. Azimuth is localization in the horizontal plane. We use timing differences. These all depend on us having 2 ears. So we use interaural timing differences. A sound coming from this side is going to hit which ear first? This one. And then it's going to hear that one. All right. Also, interaural level differences. So I mentioned that high frequency sounds get blocked by solid objects. Is my head a solid object? Yes, and that's going to block the passage of sound. So it's going to hit this ear at a higher level. It's going to be a bit quieter in that ear. So we use a combination of those two things. Elevation, we can use pinna cues. We call that a head-related transfer function. And then distance, we use a number of clues for that. One of them is the relative, so when you hear my voice right now, you hear the direct path followed by a reverberant path. And the ratio of those two will give you a clue of how close something is to you and how far away it is. Okay, that's either the air conditioning or we're in a cyclone. Okay, so we actually use, in the horizontal plane, we use both of these clues, interaural time differences and interaural level differences, and it was Lord Rayleigh in 1907 which proposed the duplex theory. It says we do both of those, but they don't deal with all the frequencies simultaneously. So interaural time differences we use for low frequency sounds. Interaural level differences are most useful for high frequency sounds. This is for sinusoids. For complex sounds like my voice, we use a combination of all of these things. So here's an illustration of interaural level differences. Let's start with the low frequencies. As I move it around in azimuth, is there much of a change in the sound level of a 200 hertz tone to my head? The answer is no. It goes up a couple of dB when it's right in front of us, but that's about it, right? Pretty flat. But when I go to a high frequency, look at this. I've got almost a 20 dB difference between sound that is coming in my right ear versus in front of you or behind you. And that's due to the fact that my head is blocking those high frequencies a lot more. So there's a great big difference between the ear that's closest and the ear that's furthest away. So can you see from this graph, high frequencies, there's a massive interaural level difference. Low frequencies, there's hardly any. That's the take-home message there. Excellent. All right, there it is there. Here's an illustration of just as a torch casts a shadow, so do high frequency sounds. And we call that the head shadow effect. All right. And they are processed by a part of the brain in that auditory pathway, which is called the lateral superior olive. It's part of the superior olivary complex. So I mentioned that each of the different bits of the auditory brainstem have a different job. This guy's job is interaural level differences. We also have interaural timing differences. So the speed of sound is a fixed number, depending on the temperature and the pressure, all that sort of stuff. It's about 340 meters per second, and your ears are a certain distance apart, and that means that sound doesn't hit both of your ears simultaneously unless it's directly in front of you or directly behind you or directly above you or directly below you. But if it's anything off to the side, it's going to hit one ear, and then a little bit later it will hit the other ear. And your ability to and it's not a straight line. Your head is in the way, right? So I've got to measure to here, and then I've got to measure from your nose and follow the curve of your head around to get the true distance, the difference in the distance of the two paths. And look at the difference in the timing. We get the biggest interaural time difference when it is exactly 90 degrees off to the side. It's going to hit this ear, and then that's the longest time gap between hitting the next ear. But any other part, it's going to be less. What is it when it's directly in front? What's the time delay between the two ears? 0. And what is it when it's 180 degrees right behind you? Also 0. Excellent. All right, and these are processed by the medial superior olive, just a different part of that superior olivary complex, whose job is to say what's the level difference between these frequencies. Mainly works for low frequencies because the wavelengths are much bigger and we can better calculate those timing differences. Our nerves are able to fire fast enough to measure those differences in the timing there. Anyone got any questions about those two? Right, which is best at low frequencies? It's this one. The timing one. Which is best at high frequencies? It's the head shadow thing. Your head's blocking the high frequencies. That's the interaural level differences. All right. We do get this effect, though, with interaural time differences, and that is if I take a particular frequency of tone which exactly matches the diameter of your head in terms of the wavelength, you could be confused by if the sound is coming directly from the right and you've got a 600 millisecond interaural time difference, this peak will now line up with that peak. And at that exact frequency, it won't sound like it's coming from one ear. It'll sound like it's coming from directly in front of you. And you've lost that localization ability only at that particular frequency. So what do you think we could do about that? is our head attached to our whole body and we're frozen in a chair? No, we can just move our head. So if you just move your head, that's the solution. And now you can tell a bit better where the sound is coming from. So all of these clues interact with the fact that we are constantly in motion and moving our head around and that sort of thing. All right, so here's a setup from our lab of testing directional hearing, playing high frequency sounds out of different speakers and having the person point to it, and we could tell whether someone has the ability to tell front from down, left from right, up from down, front to back, and so on. And I mentioned in that very, very first lecture the idea that the particular pattern of bumps and wiggles in your ear interact in a very specific way to tell you where a sound is coming from. And we call this the head-related transfer function, or head-related transform, or a pinna transform. And I mentioned that these are almost unique to each person's ear. Your brain is born, like you are born knowing one ear from the other ear. All that circuitry is in place. You can tell left from right. But your ear has to learn the particular patterns of shapes and the way that filtering depends on the direction of sound. So you will learn that, oh, a sound from here sounds slightly different, and I look up and I know that that's where it is, and I've locked in that sound changes to be like that when it's directly in front, and I learn that. And you learn that pattern, and that helps you resolve those up, down, front, back confusions. Does that make sense? So if you just had two holes on the side of your head, like a snake, you could tell left from right, but you couldn't tell up from down or front from back without this mechanism. All right, and that's a similar thing. You know how I was wearing my glasses and now things are fine? Same thing is I could get let's say you're an extra in The Hobbit and you put some sort of elf prosthetic on your ear, and now your ear bumps and wiggles are quite different, and you wear that for a few months, your brain will adapt, and now you can tell where things are coming from because your brain has relearned those clues. You've got that plasticity effect happening there. Okay, and poor Vinny here, he's a little bit troubled in that regard. I am going to put a demo on Learn. I can't do it in this environment because you need two ears to hear different signals. So you'd all need to be wearing headphones to hear this effect. But this is such a cool representation of exactly what's happening there. With Rob Patuzzi, who was my PhD supervisor, we were sitting down in the lab, and you know these things, these little lapel microphones, this guy right here, we got one and we bent it, the wire, so it was poking back out, and then shoved it down his ear. So the microphone was pointing out. So the whole thing was down his ear canal, and it was making use of his pinna was filtering the sound that was hitting the microphone. And then we got a speaker and sat the speaker there making white noise, shh, and then got him to just like rotated him on a swivel chair, and it was the equivalent of the sound moving around his head, right? So, and then we just recorded that. I can play it for you now, but when you listen with headphones, the effect is amazing. You listen with headphones and close your eyes, you can hear the sound here, and the sound will actually do a complete movement around your head like that. It's remarkable. This is what it sounds like. And that's a really cool demonstration. The other thing I'm going to post to you is a link to there was a hearing aid brand, and they were selling directional microphones and trying to sell up the spatial awareness features that you get from your particular hearing aids. And so they did this demo, but the demo itself is really cool. And what they did was had a mannequin with pinners and two microphones. And then they had, it's somewhat racist if you're Italian, but they had these Italian barbers with really exaggerated accents. Now we're going to cut your hair. And the barber comes up and we take a little off here and a little off, and he's walking around you, and he's got like a plastic bag and it scrunches, and you can hear the scissors and the shave, and you get shivers up and down your spine because you honestly feel like there's someone walking behind you, and there's a guy off in the corner playing guitar. And anyway, I'll put a link to that there as well. That's another thing to listen to with headphones. It's very, very cool. Okay, now, any questions before we finish up? It's a lot of content there. You may be frightened. There's no need. I've also posted a list of ILOs, and that really strips it down to just these bullet points. Just know the difference between this and this. What does this do? It's very, very clear when you have a look on there, and we can have sessions where we go over them. If you've got any questions, post it on the forum. I'll help you. Come to my office. I'll sit down and talk you through all of this. I love talking about this. Okay. Just before we go, I might have mentioned that I also play music, and I like making songs and things like that. This particular tissue, the Stria vascularis that I mentioned, is a 3-cell layer pumping epithelium. Its job is to pump. It pumps that potassium, so it creates that high potassium concentration in scala media, and it creates the 95 millivolt endocochlear potential, which powers the whole active process. That's the inner ear battery for all of this. So I wanted to drill into my audiology students the importance of this tissue and get some of those numbers in terms of its driving potential. So the driving potential is the difference between what is the voltage difference that's making the potassium flow into these hair cells, and the difference between plus 95 millivolts and minus 70 millivolts is like and this one here, the inner hair cells, is like 135 millivolts, right? So I needed to get all of these numbers into their heads, and it was April Fool's Day, and so I recorded a song about Stria vascularis, the bloody stripe, and it's called Bloody Stripe, and it just drums in these numbers. So would you, yeah, we've got a few minutes. Shall I just play it on your way out? All right, are you ready? Here we go. So this is the only punk rock song about auditory physiology that contains in-text references as well. So I do hope you enjoy it.